Hello friends. So the next INICET is just round the corner. You barely have 2 months for the INICET, and all of us know that there is a lot of repetition, at least for the topics in the INICET exam. And in this paper, typically anywhere between 10 to 15 questions will be there from biochemistry. And the type of biochemistry which comes in INICET is slightly different from what you get in the NEET PG exam. So in this video, what we're going to do, we'll take a quick look at the biochemistry questions from the last 3 papers. Okay, so 2 papers of 2022 and the November paper for 2021. I have aggregated the questions from those 3 papers, and we'll take a quick look without spending a lot of extra time. Just the highlights for the topics from which the questions have been picked, and it will help you to quickly revise the important topics, and it will also reorient you about the topics which are being asked in the INICET, in contrast to what we see in the NEET PG. Obviously, I told you there's some differences. So once you go through this video, you will be familiar with the focus areas for the preparation of the INICET, so that you can quickly do your revision and you can make your revision more effective in terms of the time management. Okay, so let us quickly take a look at the questions in the last 3 paper of the INICET exam. Okay, so first question: which of the following consumes the highest energy during assimilation? You have protein, carbohydrate, MUFA, PUFA. So this is a common theme in the INICET exam. We see very frequently they will talk about the specific dynamic action of the food. They will talk about the respiratory quotient of the food. They will talk about the definition of the respiratory quotient. So these are some of the common aspects which they discuss for the nutrition. Okay, so the highest energy during assimilation is what we call as specific dynamic action, meaning the amount of energy that the body spends to assimilate a food substance. That is the specific dynamic action. Okay, and specific dynamic action among the food materials, among the macromolecules, will be highest for the proteins in the range of 30%. Okay, it is highest for the protein in the range of 30%. For carbohydrates, it is the lowest in the range of 5 to 10%. For the fatty acids, anywhere between 10 to 15% would be the energy, relative energy, which is spent to assimilate the substance. So remember these points. In the specific dynamic action, take a look at the RQ of the different substances and the explanation of the specific dynamic action. Then, which of the following is not amphipathic? You have the molecules glycolipid, phospholipid, sphingolipid, triacylglycerol. So this is another type of question they'll ask. They ask something about the physical nature of the molecules. You can think of it as the biophysics part of the biochemistry, where they try to see whether you understand the macromolecules with which you are dealing in biochemistry or not. Okay, so this amphipathic nature becomes very, very important when we talk about the membrane structure, particularly if you're talking about the lipoproteins, because the membrane, you know, has to interact with both the water and the inner content, and in case of lipoproteins, the inner content is typically the non-polar molecules. So there the amphipathic nature of the molecules becomes very, very important. So in this context, you have to remember that the molecules, the glycolipid, phospholipids, sphingolipids, these are amphipathic, but the triacylglycerol is neutral, or you can say non-polar, strictly neutral or non-polar. It is commonly found in the core of the lipoprotein. If you look at the lipoprotein structure, it will have the 2 layers. It will have the 2 layers. The outer layer is what is made up of the amphipathic molecules, whereas the core is made up of the non-polar component. This is how the non-polar lipids are transported in the body. Okay, so the lipoprotein structure is a specialized structure to transport the non-polar lipids. Otherwise, the lipids will not be able to travel in the blood. They are non-polar, they will not be able to travel in the blood. So they need a structure which encloses them, and it is transported in the blood. So the molecules at this side would be having the hydrophobic nature. The molecules towards the outer side are having the polar nature. So because the same molecule is constituting the layer, the molecules must be amphipathic, meaning they are at one side they are polar, or the other side they are non-polar. Okay. Wobble hypothesis explains which of the following concepts of the genetic code. Your options are degeneracy, and ambiguous, non-punctuated universality. So another aspect, they talk about the basic concepts in biochemistry, particularly in molecular biology. So the concept is an important emphasis in the INICET exam, which is not there in the NEET PG exam. In NEET PG exam, they will mostly focus on the applied part, but in INICET, some focus is there on understanding, whether you know the basic underlying concepts of the various topics that you're discussing or not. Okay, so Wobble hypothesis, what it says, at the third position on the codon, the interaction with the anticodon is flexible. Okay, so the same nucleotide on the anticodon is able to interact with two different nucleotides on the codon. Because of this, what happens that for the same amino acid, see the tRNA is attaching one amino acid, but now it is able to interact with two different codons. So for same amino acid, we have multiple codons. This is the property of degeneracy. Okay, so together the Wobble hypothesis and degeneracy explain what is known as the silent mutation. So what happens in silent mutation? There is a change in nucleotide, but there is no change in the amino acid. Reason being that we have multiple codons for the same amino acid. So if the change in nucleotide results in another codon which is coding for the same amino acid, then there will be no change in the protein. This is what is silent mutation. So keep in mind, the Wobble phenomena and the degeneracy are the responsible factors for the silent mutation. A change in the nucleotide doesn't result in a change in the amino acid in the translated protein because of these two factors. So Wobble hypothesis explains the degeneracy. Together they are responsible for what is known as silent mutation. In spite of change in the nucleotide sequence, change in the amino acid sequence in the protein is not seen. Then we come to question: a patient with thiamine deficiency develops acidosis. Which of the following enzymes was affected? The options are pyruvate carboxylase, pyruvate dehydrogenase, Pyq, and lactate dehydrogenase. So here the examiner is trying to see your knowledge about the composition of the functional molecules. In this case, the examiner wants to see whether you know the requirement of thiamine is there for which of the enzymes, right? In contrast, if you go to the NEET PG exam, they will rather want to know that a clinical condition is associated with the deficiency of which vitamin, okay, or which enzyme can be used to identify the deficiency of a particular vitamin. Rather than looking at the composition of the enzyme complex and requirement of the different vitamins, they would rather focus on the clinical aspects more. So here, patient with thiamine deficiency develops acidosis. So now you have to tell us in which of the following enzymes the thiamine will be required. Lactate dehydrogenase doesn't require thiamine. Pyq carboxykinase will not require the thiamine. The pyruvate dehydrogenase, yes. Pyruvate carboxylase rather requires the biotin. It is one of the 4 carboxylases. All the carboxylases will require the biotin. So a correct answer here would be pyruvate dehydrogenase. Please note, pyruvate dehydrogenase requires vitamin B1, that is thiamine. It requires B2, that is riboflavin. It requires B3, that is niacin. It requires B5, that is pantothenic acid. And it requires the lipoic acid. Okay, so the 5 coenzymes are required by the pyruvate dehydrogenase, the alpha ketoglutarate dehydrogenase, and the branch chain keto acid dehydrogenase. So 3 enzymes: the pyruvate dehydrogenase, the alpha ketoglutarate dehydrogenase, and the branch chain keto acid dehydrogenase. All these 3 enzymes are complexes. They are made up of 3 enzyme units and 5 coenzymes. These 5 coenzymes are common in each of these 3 enzymes. Okay, remember this point. Next question: which of the following tests will be positive in the urine of a patient who has undergone a prolonged starvation? So now you see the biochemical tests which are done in the lab. Again, more commonly asked in INICET in comparison to the NEET PG exam. In the NEET PG exam, they will rather give you the results and ask you to make the diagnosis. Here it's the other way around. They've given you that the patient is having prolonged starvation. Then what are the likely findings in the urine? Which of the following tests would be positive? Okay, so in this case, what you have to understand, when the patient has prolonged starvation, okay, in this case, glucose level is low. But what happens, there is increased in the ketone bodies. There will be increase in the free fatty acid also, but it doesn't come in the urine. Ketone bodies, however, will come in the urine. Okay. And now look at the tests. The first test is obviously the Bendick test, which is for reducing substance. Glucose is not there, so it will be negative. Second test is the Rothera's test. It is for the ketone bodies, so this one should be positive. The third test is for the proteins. Okay, so this will also not be positive until there is some inflammation or there is some chronic kidney disease. You don't expect the protein to come out in the urine. So in this patient, the protein will not be there. Only the Rothera's test will be positive, that is for ketoacidosis. So in starvation, we get the ketoacidosis. Along with starvation, you also get ketoacidosis in type 1 diabetes patients, that is diabetic ketoacidosis, and in alcoholics. These are the three common conditions where you are going to see the ketoacidosis: the starvation, the patient of type 1 who lands in diabetic ketoacidosis, and in the chronic alcoholics, you are going to see the ketoacidosis. Obviously, if it were a patient of DKA, then the glucose would also be there, and if the patient also has the concomitant the chronic kidney disease, then the protein can also be there. In that case, all the three tests might be positive in the patient. Again, you can see most widely accepted method for HbA1c estimation is ion exchange chromatography, affinity chromatography, immunoturbidimetry, capillary electrophoresis. Focus on the technique, on the basic concept. Okay, focus on the test. That is one important feature of the INICET exam. So here you have to know the answer, that the HPLC based on the ion exchange chromatography is the preferred, or you can say the gold standard for the estimation of HbA1c. The HPLC based on ion exchange chromatography, that is the gold standard for HPLC. The other methods can be used, but they are not the gold standard. Now, which of the following is true about mitochondrial DNA? You have various options. Higher rate of mutation as compared to nuclear genome, double stranded circular DNA with 16,000 nucleotides, slight change in nucleotide sequence compared to cellular DNA. Mitochondrial DNA encodes all etc proteins, and then we have the combinations 1 and 2, 2 and 3, 1 and 3, 2 and 4. So here you have to understand two things. Number one, the question on mitochondrial DNA, very frequently asked in the INICET exam. Please look at the very short note that we have on the mitochondrial DNA. All right, you are always going to get one question from mitochondrial DNA, the inherited disorders, the inheritance pattern. That should be clear in your mind. Second thing, the options which are there, some of them are ambiguous statements in the sense it is not strictly wrong, but it is not strictly correct also. It is close to right. So you have to look at the options and choose the best possible answer. Okay, so if you look at, let's say, option 2, double stranded circular DNA with 16,000 nucleotides, this is somewhat right but somewhat wrong also. Why? Because number of base pairs is 16569 base pairs. Okay, so this should be 16.5 but it will depend. We have to choose two of them. If none of the others are correct, only one other is correct, then we have to go for this. But if you have a better option somewhere else, then you can skip this, because strictly speaking this is not correct. Okay, so this is another way in which the questions will be there in the INICET. You have to choose the best possible answer by looking at the available options. Generally this type of ambiguity will not be there in the NEET PG exam. So you have to be careful when you're doing the INICET. You have to keep your eyes and mind open. Read all the options before you mark the answer. If you look at option 1 and you think that is the answer, don't directly jump to mark the answer. Read all the other options. Maybe there is one better option than that particular option. Okay, so please be very careful. In INICET you might run afoul of a better option which you have not read in the question. Okay, higher rate of mutation as compared to nuclear genome. Okay, correct. Slight change in nucleotide sequence compared to cellular DNA. Now, this is also slightly ambiguous. Reason being, there is no change in nucleotide sequence, but the interpretation, that is the translation which is there, that is slightly different. Okay, so in certain cases, mitochondrial DNA encodes all etc protein. Again, wrong. So, out of these two, we have to choose one, which is the better statement. I would say that double-stranded circular DNA with 16,000 nucleotides is better. There is no change in the nucleotide sequence. If the option said that there is change in the translation of the codons, then I would go for that. Obviously, this is a recall question. Maybe the statement was like that, but in the current scenario, I would prefer to go with 1 and 2. In this case, I would go with 1 and 2. If the option 3 was slightly more clear, that the translation of the codons in the mitochondrial DNA is somewhat different from the nuclear DNA, then that would be a better, more clear-cut answer. Okay. Next question, which of the following is associated with defect in formation of collagen? Now, this is something which you can see in both types of exam, NEET PG and the INICET. INICET generally gives less images, NEET PG will give you more images. So, recently in the NEET PG exam, we saw that similar question was there, and the images can be there. Okay, so obviously we know that collagen synthesis requires two things. It requires vitamin C and copper. Okay, so and when the collagen synthesis is defective because of particularly the vitamin C deficiency, in that case we get the scurvy. When there is defect in collagen synthesis due to the vitamin C deficiency, you get the scurvy. In rickets, there is defective mineralization. In beriberi, there is thiamine deficiency, because of which various the enzymes of metabolism are affected. And in pellagra, there is the deficiency of the niacin. Okay, there is deficiency of niacin. Next question, which of the following test is best for quantitative analysis of RNA samples? Again, you can see focus on the techniques, the test, the principle. That is very peculiar for the INICET exam. Real-time PCR, reverse transcriptase PCR, PCR with restriction fragment length pair technique, reverse transcriptase followed by real-time PCR. Okay, which of the following test is best for quantitative analysis of RNA samples? Okay, now in this case, real-time PCR is strictly speaking not for RNA, it is for DNA. Either whether it is the complementary DNA or it is the double-strand DNA, doesn't matter. The reverse transcriptase followed by reverse transcription followed by PCR would be a good answer, but let's see, look at the other options. PCR with restriction fragment length polymorphism, no, this is used to identify the single nucleotide polymorphism, so this is not for quantitative analysis of RNA. Reverse transcriptase, rather this should be reverse transcription followed by real-time PCR. Now this is the best and the most suitable answer, because in real-time PCR we can do the real-time PCR, also known as the quantitative PCR. So quantitative analysis of the RNA can be done when you follow the reverse transcription by the real-time PCR. So first you convert the RNA into cDNA, then you use the cDNA as a template for the PCR, and by performing the qPCR, real-time PCR also known as qPCR, quantitative PCR, you can do the quantitative analysis of RNA samples. So D would be the best possible answer. Moving to question number 10, what is the repair mechanism associated with the CRISPR-Cas9? Again, you see the technique. I'm repeating the concepts, the basics, the tests, the techniques. That is more in focus in the INICET exam. Your options are non-homologous end joining, the mismatch repair, base exchange repair, and nucleotide exchange repair. You should know that the CRISPR-Cas9 is based on the non-homologous end joining. We introduce a cut, the change in the sequence, and the joining of the two strands. That is how the CRISPR-Cas system will work. Okay, Moving on, DNA protein interaction is studied using, again you can see technique is mentioned, DNA footprinting, DNA fingerprinting, ELISA, northern blotting. Okay, northern blotting is not for DNA, ELISA is only for protein, DNA fingerprinting is only for DNA, DNA footprinting includes both DNA and the protein. Basically where the protein is there, in that part the amplification of the DNA will be decreased, so that can be used for the DNA protein interaction. And this is sometimes also known as southwestern blotting. Okay, so the DNA footprinting or the southwestern blotting can be used to read the DNA protein interaction. Okay, which of the following type of inhibition is depicted in the following Lineweaver-Burke plot? Competitive inhibition, non-competitive inhibition, uncompetitive inhibition, and suicidal inhibition. Now, this is one of the very frequently repeated questions in both the exams, the INICET and the NEET PG exam, talking about the different graphs and asking you to identify the type of inhibitions shown in the graph. Sometimes they can club it with the pharma, and there they can ask you the drugs which will act by the mechanism shown in the graph. So you should be careful about this particular question. Now here I give you the very simple trick how to identify the different inhibition graphs. Remember, the Vmax is normal in some cases and the Km is normal in some cases. Now you also have to understand that on the Lineweaver-Burke plot, the point where graph touches the y-axis, that is 1 upon Vmax, whereas the point where the graph touches the x-axis, that is -1 by Km. What this means, if the Vmax is same, the point here will overlap. If the Km is same, then the point over x-axis will overlap. Okay, so in that case your graph would be something like this. Okay, so looking at the common point, you can make the interpretation. Remember, the Vmax is normal in case of competitive inhibition, Km is normal in case of non-competitive inhibition. This is by definition. If none of the 2 points are common, then you're looking at uncompetitive inhibition. So just with the help of what is normal, you can quickly identify the graph, the type of graph inhibition shown in the graph. In this case, you can see that the common point is on the y-axis. The Vmax is a normal. Vmax is normal, meaning we are looking at the competitive inhibition. Please remember, the Lineweaver-Burke plot is also known as the double reciprocal plot. So you can get the same question where they'll just replace the name of Lineweaver-Burke plot with the double reciprocal plot. Don't get confused. A patient with Maple Syrup Urine Disease, all of the following amino acids should be restricted in diet except methionine, valine, leucine, isoleucine. Now first you have to understand Maple Syrup Urine Disease, what is happening here. There is defect of the branch chain keto acid dehydrogenase. Okay, so branch chain amino acids, that is what you have to restrict. Which are the branch chain amino acids? Branch chain amino acids are valine, leucine, isoleucine. Valine, leucine, isoleucine. These have to be restricted. Methionine doesn't need to be restricted. Okay. Question number 14. All of the following are true about Human Genome Project except took nearly 2 decades, total genes 20,500, next generation sequencing used, took international collaboration. Now you can look at this question in 2 ways. Number 1, this is one of those questions which you are not expected to know, and if you answer it, it will at best be a guesswork. However, if you know few points about this particular topic, then you might try to eliminate the options which are not correct or which are not likely to be the answer. So either you can think of it as a question which you can let go, or you can think of it as a question where you might try to eliminate the options which are not available. Personally, I would say this is when this question was asked, I didn't expect any of my students to answer it correctly as per their knowledge. Okay, because the very precise options were there which it is not expected from the student. Okay, took nearly 2 decades, correct. Total genes 20,500. Once the whole process was completed, this was the final conclusion drawn by the Human Genome Project. Very correct. Next generation sequencing used is not correct. Next generation sequencing developed as a result of the Human Genome Project at the time when we started the Human Genome Project, and when the first phase was completed in around 2004 to 6, at that time next generation sequencing was not well developed. Took international collaboration. Very correct. So in this case, next generation sequencing was used is not correct. Okay, please keep that in mind. In Sanger's sequencing, see again, technique. The INICET is talking so much about concepts, tests, techniques, the underlying concepts that you have to know. In Sanger's sequencing, DDNTP is dideoxy nucleotide triphosphate are used because of which of the following properties? DDNTP exhibit higher affinity for DNA polymerase? No. DDNTP exhibit lower affinity for DNA polymerase? No. These two are not the features for using it. Insertion of DDNTP results in prevention of elongation of chain by DNA polymerase? Yes. DDNTP causes chain elongation by DNA polymerase? No. So see what happens. C1, C2, C3, C4 and C5, right? So here you have the phosphate, and the new nucleotide will come and get attached at C3 via the hydroxyl group. Normally the C2 is deoxygenated, the hydroxyl group is not there. That is the deoxynucleotide triphosphate. When I say dideoxy, the hydroxyl group at C3 has also been eliminated. That is why the next nucleotide which comes in is not able to attach, prevents the elongation of chain by DNA polymerase once the DDNTP is incorporated. So wherever the DDNTP will get incorporated, chain elongation will stop. But we can refer the position of the type of nucleotide at that place, which will help us to read the sequence of the DNA. So in Sanger sequencing, the use of DDNTP is to prevent the further elongation. Wherever the DDNTP will get incorporated, further elongation will stop. You are expected to produce a protein of interest. Arrange the following steps in the correct order for the same. Your options are lysing a bacterial cell, incorporating cDNA into bacteria, SDS page, protein elution, hybridization, and then the various combinations are given. So this is the part of steps of genetic engineering, how we do the genetic engineering and produce a desired protein. As it is mentioned in the question, you are expected to produce a protein of interest. So basically what you have to do, you identify the gene, you add the gene into the bacteria, you allow it to get incorporated, then it will get expressed. Once it is expressed, you lyse the cell, you separate the protein from all other proteins, and then you extract the pure form. All the steps are not mentioned here. Okay, please note, all the steps are not mentioned here. So let's look at the options. Gene, mention of gene is not there. Second, incorporating into bacteria, okay, this is there, so this should be our step 1. Then the incorporation or hybridization is mentioned, that should be our step 2. Then there is the expression, it is not mentioned in the options. Then there is lysing of the bacterial cell, it should be our step 3. Then separation of the protein, which will be done by SDS page, and the pure form, that is by protein elution. So 2 should be first. Okay, 2 should be first. Then we should have 5. So we have 5 here. Let's see if other options are in order. Okay, 1, 3, 4. So this is the correct sequence as per the given options. Please note, all the steps which are required in this genetic engineering are not mentioned to prevent complication, but it becomes confusing also because all the steps are not there. Again, one of those questions where if you're not sure about the genetic engineering, it would at best be a guesswork. But at the same time, focus on the technique. Genetic engineering, very common technique for producing a desired protein. Misincorporation of uracil into DNA is seen in deficiency of B12, folate, B6 and vitamin E. Again, this is a conceptual question where you have to understand which of the vitamins is required for conversion of uracil into the thiamine. So you have to remember that DUMP gets converted into TMP, where the methylene tetrahydrofolate is required. Okay, meaning if the tetrahydrofolate is not there, the TMP formation will not take place, and this can get incorporated into the DNA in place of the thymine. So which is what is mentioned in the question. So your answer should be folate. Folate deficiency can result in misincorporation of uracil into the DNA. Now, continuing with the same vein, deamination of methylated cytidine residue results in incorporation of which of the following into the DNA? So now you have to look at it in different ways. First, if you have the cytidine and you do the deamination, in that case you will get the uracil, right? Now, if you do the methylation, of uracil, we just mentioned, it gets converted into the thymidine. Okay, so deamination of methylated cytidine result in formation of thymidine. It will result in formation of the thymidine. Match the following transporters with the molecules that they carry. Obviously, this is a recall question, so there's some difference in the question which we could not find out. Please note, all of them are correctly matched. Okay, icosanoids are formed from, again, the very basic concept. Icosanoids are formed from the carbon 20 polyunsaturated fatty acid. In this case, it is the arachidonic acid. Arachidonic acid is a precursor for the icosanoids. Icosanoids are very important. They give rise to a large number of functional molecules. The thromboxanes, the leukotrienes, the prostaglandin, the prostacyclins, all of these will come from the icosanoids. RBC uses which of the following substance during fasting starvation? Please note, RBC can only use glucose, because other fuels metabolism requires mitochondria, which is not there in RBC. So about RBC, there is no ambiguity. It is the only cell which exclusively uses the glucose. So you can just simply mark glucose without any confusion. For the brain, it is mainly glucose. When I say mainly glucose, it means that in starvation it can use significant amount of ketone bodies, but otherwise it will mainly use the glucose only. Okay, question number 22. RNAi acts through. So when I say RNAi, it means we are talking about the microRNA or the siRNA. Okay, the microRNA or the siRNA. The microRNA or siRNA acts through what is known as RNA interference. That is why it is written as RNAi. The result of RNA interference is knockdown phenomena. Less amount of the protein is formed in spite of the RNA being formed. Okay, because either the RNA is degraded or it is destabilized or the translation is prevented. Because of all these reasons, the amount of protein which is formed is decreased. This is called the knockdown phenomena. Please keep in mind, the knockout and knock in, these changes will occur at the level of DNA, whereas the knockdown phenomena is something which is happening at the level of the messenger RNA. The knockdown occurs at the level of messenger RNA. Knockout means you take out a segment of the DNA. Knock in means you add a segment of the DNA into the gene. Methyl tetrahydrofolate accumulation and functional deficiency of folate is seen in the deficiency of. What is described in the question is actually what is known as folate trap. Folate trap is seen in case of deficiency of B12. Okay, so the answer in this case is B12. B12 deficiency will result in folate trap. The folic acid will get accumulated in the form of methyl tetrahydrofolate, and it cannot be used up. Why? Because methyl tetrahydrofolate first has to get converted to tetrahydrofolate, for which B12 is required, and then it gets converted to the functional forms, the methylene, methinyl, formino, formyl. All of these are formed from the tetrahydrofolate, not from methyl tetrahydrofolate. So conversion from methyl tetrafolate to tetrafolate requires B12. If B12 is not available, the folate will get trapped in the non-functional form. This is what we call folate trap, and in these cases you will have the features of B12 deficiency as well as the features of the B9 deficiency, that is called the pernicious anemia. Insulin inhibits which of the following lipase enzymes? Similar question has been asked in the NEET PG exam also. So you should remember the insulin inhibits the hormone sensitive lipase which is commonly present in the adipocytes. So as long as insulin is there in circulation in abundant amounts, the triglycerol in the adipocytes will not be mobilized. Once the fasting and starvation sets in, the insulin level starts falling, this inhibition will be lost. That is when the hormone sensitive lipase will become active and it will start mobilizing the triglycerol, the fatty acid which is present in triglycerol, which goes into the circulation, tissues will take up, liver will take up, liver will convert it into ketone bodies also. Glycosylated HB is best detected by, this is a repeat question as we can see, and we have already said the ion exchange chromatography using HPLC is the best method, gold standard method for identifying the HbA1c. Which of the following methods uses RNA? Repeat question. We already said the RT-PCR uses the RNA Western blot, will make use of protein. Sanger method is for protein and the DNA. The G banding is for DNA. Cytochrome C oxidase is inhibited by all except, remember, cytochrome C oxidase is inhibited by H2S, carbon monoxide, cyanide and azide. Methane is not an inhibitor of cytochrome C oxidase. Please also note cytochrome C oxidase is more commonly identified as complex 4 of the electron transport chain. This tells you that knowing the number of electron transport chain is not enough. You should know the names of the complexes also. What is the name of complex 1, 2 and 3 also. Please note that the F0 F1 particle is sometimes also described as complex 5. So in the question if you see complex 5, please don't get confused. We are talking about the F0 F1 complex or the ATP synthase. Binding site of microRNA on messenger RNA. This is again that question which we don't expect you to know. The correct answer is the 3' untranslated region of the messenger RNA. The end point and the part which is not getting translated, that is why we call it UTR, the untranslated region. This is where the microRNA will interact with the messenger RNA. It will look for homology, and if homology is there, then it will prevent the translation of that messenger RNA. Which of the following enzymes diagnostic marker is incorrectly paired? Let us see. Lipase with pancreatitis, this is correctly matched. High LDH1 LDH2 in myocardial infarction, this is correctly matched. This is known as flipped LDH ratio. Ceruloplasmin in hepatoventricular degeneration, Wilson's disease, this is correctly matched. Beta glucose cerebrosidase in von Gerkes, no, this is not correct. In von Gerkes, the deficient enzyme is the glucose 6 phosphatase. The beta glucose cerebrosidase is deficient in the Gaucher's disease. Okay, so the enzymes and the associated disorders is a common question. Which of the following is not used for protein precipitation? See, again a technique. Heavy metals, yes. Alcohol and acetone, yes. Change in pH other than isolytic pH, no. Change in isolytic pH is used for precipitation. And trichloroacetic acid, yes. So the option is C. What is seen in G6PD deficiency? Your options are decreased membrane lipid peroxidation, decreased generation of reduced glutathione, decreased NADH generation, decreased scavenging of RBC of macrophages, and hemolysis. Again, it's a recall question, so we had some confusion about which of these two options were present. Please note, decreased membrane lipid peroxidation. Not really, because the NADPH and glutathione are mainly preventing the protein oxidation, you can say, and they are acting on the peroxides, so they are not you can say lipid soluble antioxidant per se. There the better role of vitamin E is there. Decreased NADH generation, no. Decreased NADPH generation is there. Decreased scavenging of RBC by macrophages. Macrophages don't scavenge the RBC per se. So this is and moreover, this scavenging is not dependent on production of NADPH. Rather, the macrophages require the NADPH for the oxidative burst, and that is for scavenging of the bacteria, not for the RBC. Decreased generation of reduced glutathione does happen, and hemolysis of the destruction of the RBC does happen. So these are two possible options. So the first better answer is reduced glutathione, and hemolysis you have to see which options is present in the exam. Both of them are correct. Next question: which of the following supplement is required more for a lactating woman than the pregnant female? All of you know that vitamin A is required in larger amount during lactation than during the pregnancy. Sideroblastic anemia. Sideroblastic anemia we already mentioned is sideroblastic anemia is seen in the deficiency of vitamin B6, that is pyridoxine. So here I'll quickly tell you the different types of anemia and that you may come across. You get the normocytic normochromic anemia. This will be seen in A. riboflavinosis. Normocytic normochromic anemia in A. riboflavinosis. Sideroblastic anemia, it will be seen with the pyridoxine deficiency. Okay, then you get the megaloblastic anemia. This will be in B9 deficiency. Then you get the pernicious anemia. Pernicious anemia, this will be seen in the B12 deficiency, where in addition to the megaloblastic anemia we also see the neurodegeneration. Neurodegeneration is another feature. And lastly, the iron deficiency anemia, this will be seen with the vitamin C deficiency. Why? Because vitamin C is required for the successful absorption of the elemental iron, not for the heme iron but for the elemental iron. So if vitamin C is not there, absorption of iron will be decreased, it will not be effective, and these patients are likely to suffer from the iron deficiency anemia. So the 5 different types of anemias which are associated with the different vitamin deficiency. So these are the important questions which were asked from biochemistry in the last 3 papers, and you can expect the questions from these associated topics to be repeated again. So obviously you have to cover the main part of the syllabus, but these 33 questions and these 33 topics are going to give you a leg up in your preparation for the INICET in May. Please go through this video and try to revise the content. If you have any doubts or queries, don't hesitate to write in the comments. I'll get back to you, and please like and subscribe for more videos like this. Thank you very much.